in theory, final talk and the third plenary of our meeting. And I'm very happy to welcome Kathrin Ohla here, who I hope is there and could start putting up her slide.
I am.
Yes, wonderful. Her background is in psychology, and in her research, she aims to understand how the human mind senses, experiences, and processes taste, and in particular, how this processing connects to behavior. In terms of her career, she has held various positions as researcher and professor and research group leader in research centers, in academia and also in industry, and as well as in universities, and that was in Germany, in Switzerland, in Philadelphia. So she has been kind of around the globe in a sense, and she has received a number of awards, and in particular, just this year, the so-called IFF Award for research in the psychophysics of human taste and smell. And in the last year and a half, I guess she was very busy also organizing research and into and about COVID-19 and the loss of smell and taste that it induces. However, this is not what she's going to talk about today. She's talking about taste. I don't know the actual title, which is very embarrassing. Sorry about that. And before you begin, just one note. In one of the previous channels, there was a problem with a microphone. So after this last plenary talk, we will open channel 2 again for the talk by Nadia Balmabrook, which did not take place in the previous channel. So don't go away after Kathrin speaks, and we have also done our closing remarks. There will be another talk in this session, well, in the other session actually. So please, I give you the opportunity to talk, please.
So thank you, Evelyn, for this very nice introduction and for preparing everybody for this shift of gear topic-wise that is supposed to come. So let me try share my screen, which should work now. Can you see my slides or my presenter view?
I see two small kind of blocks.
Okay, let me try again. That is not supposed to be the one. So you should see only one. So let me try this. So now you should see only one slide. Wonderful. So the title of my talk is A Matter of Taste, Large-Scale Gustatory Response Patterns and How They Predict Taste-Related Behavior. And this is all in human participants. And I will also take the liberty to ignore the animal literature, which stems mostly from rodents, but which is beautifully aligned with our data. So when we talk about taste, normally we actually confuse taste and smell during the eating experience. But when I talk about taste, what I actually mean is really the basic taste qualities, such as salty, bitter, sweet, sour, umami, which is a yummy protein taste, maybe also fat and others. So there are only very few categories. Most of these categories people still discuss whether they're really true taste qualities or not. But salty, bitter, sweet, and sour are those that are most established and everybody agrees on, and those will be in the focus of my talk. So most of these taste qualities can be put in relation with also a physiological function. For example, think of salty. which helps us actually maintain our electrolyte balance. Just think of a hot summer day, you maybe even exercised, you've been sweating, you lost a lot of salts. So then very often you experience some kind of craving for salty food. And this is your body telling you to fill up the electrolyte stores. So, and similarly, we may crave something sweet, which is just our body telling us we need quick energy, and that typically comes from carbohydrates such as sugars. Sour and bitter, on the other hand, have a little bit of different functions. So they do also have a warning function, because sour lets us sense acids in our food, and at high concentrations, acids can be harmful to our tissue, to our health. They can literally burn our skin and damage our teeth, and also indicate maybe spoiled foods, like milk is not supposed to taste sour, for example. And bitter is also an interesting quality in a way that most bitter substances are actually toxic, at least at high concentrations. So we don't like them inherently. So, but we can learn to appreciate them. And I put here some food examples which taste bitter, like asparagus, and also beer and coffee. I mean, we learn to appreciate that simply because it has nice pharmacological side effects. The fact that probably also I'm speaking here today is that we know almost nothing about how the sense of taste works. And I was fascinated by that as a PhD student. When after my PhD, I started this research into taste. And still in this recent edition of this textbook shown here to the left, which is really used to educate our next generation of psychologists, you see the discrepancy of the amount of pages allocated to each sensory system. Like while we have 142 pages dedicated to the visual system, we have 5 pages dedicated to taste. And frankly, not everything is like really up to date on those 5 pages. So there's a tremendous need for research, and I have spent the past years to shed some light on urgent questions on that matter. But that is not my work. So in the late 70s already, people have shown actually that we have inherent preferences or aversions to those different tastes. For example, here you see snapshots from video recordings taken from newborns who were given different taste solutions. So on the upper left, you see a baby who got a sweet, and obviously an umami solution yields the same effect. So it got some sucrose on his lips, and it's making a happy suckling face. It's like smacking its lips. And on the upper right, you see the typical lemon face once the baby is given a sour taste solution based on citric acid. It's very unhappy. When it receives the salty taste shown in the lower right, it shows not much of a response. It's just like water. And on the lower left, you see the bitter response. And if you were to look at the video, you would see that the baby is crying and gaping and trying to get rid of this horrible taste. So those studies have been replicated in many species and also in humans again and again. So we can surely say that we have an inherent preference for sweet and umami, an indifference to salty taste, and an aversion as we are born to sour and bitter taste, as those may be harmful substances. And when we look actually in what breast milk is actually constituted of, we will find a high amount of sugar and also a high amount of protein. which would result in sweet and umami taste. And any newborn that wants to thrive and live does very good in appreciating those tastes. So there seems to be a very natural idea behind this here. So as we grow up, the preference for sweet remains, unfortunately for some of us, because it's very hard to resist all these delicacies that we are offered. The sour taste aversion, particularly for high concentration, remains. And salty taste is very prone to nutrition and dietary factors. So it's really a spiral. The more salt we eat, the more salt we like, the more salt we eat, and you see the pattern. However, it can be reset. It's not an endless spiral. So if you do a low sodium diet, you can reset your salty preference and start over again. And bitter taste, I had mentioned this already, this initial aversion we learn to actually get rid of and appreciate bitter substances, hopefully not only beer, but also the healthy green vegetables that contain a lot of healthy ingredients. And we can learn to get rid of this aversion and learn to appreciate that taste simply through exposure in our family. We learn our parents eat that, they are not going to die from eating the peas, so we may try as well, or eating the broccoli. So this just to give you a broad introduction into what the sense of taste is actually about. So here I'm going to present you 4 different studies today. So all of them actually tackle a similar problem. So we do have different spaces or levels that eventually lead to perception and action. So in the gustatory sense, we have a chemical space that is, for example, our sodium chloride or our common salt or sugar or citric acid. And this physical stimulus or chemical stimulus is actually coming in contact with our sensory organ, which is the tongue. And here it elicits a cascade of neuronal signals that eventually is processed in different parts of the brain. And that is our neuronal space or step of the processing. After this processing of the sensory stimulus, our brain or our minds are actually interpreting the stimulus. They are making sense of it. Because what is very important to understand is that what we call perception is really not the mirror image of what is out there in the physical world. It's maybe an approximation if we are lucky, but it can be also grossly misleading because our brains have lots of memory, we have stored lots of experiences, we have expectations, we may be distracted. So many factors in our mind actually influence at the perceptual experience that we are having when coming in contact with a physical or chemical stimulus. And depending on this interpretation, our perceptual interpretation, we may take action, like take another bite or spit it out or avoid the food next time altogether. So my research has been focusing largely on understanding how the brain activity may actually also reflect what is outside in the chemical world, what defines our stimulus material, because obviously whether we taste or experience something salty or sweet, and also the concentration of it depends on our actions, right? It's physiologically and also homeostatically so relevant. So there should be good discrimination in our brain between the different tastes and also the level of the concentrations that they are available in our nutrition. So in order to even get this project started, we had to overcome many hurdles, mostly technical nature. So you can probably imagine when we have a beverage or some kind of solution with the taste, I mean, we cannot just pour that into somebody's mouth or have them just take a big sip while we monitor brain activity. So particularly when you're interested also really in a nicely temporal resolution, you would want to use electrophysiological measures, like you can see here in this picture where the person is wearing a cap with electrodes mounted into to pick up the electrical brain activity through an EEG amplifier. So we had to think of a stimulation that can actually be controlled. Because when you just take a sip, how do you control the time of the stimulus? How do you control the amount, the motions, etc.? And think of doing this for a couple of hours. Participants may actually feel pretty full and don't want to continue the experiments. So we are using a somewhat peculiar stimulation. So, and the protocol is as follows. So to the very left, you see a precision gustometer. It's basically a very fancy syringe pump system. So it has a series of syringe pumps. Each of them are connected to bottles which hold a taste solution. And they have another valve that lets the syringe dispense the solution into this big blue hose to our participant. And along the way, we have the individual tubes in a warm water bath so that we can keep the temperature constant and near body temperature because we don't want to investigate the brain's responses to temperature cues. So once the stimulus or once the hose arrives at the mouth of the participant, it's actually eliciting very irregular spray pulses. So we basically mix a solution with high pressurized air to get an aerosol. And we spray that stimulus onto the tongue with a very irregular rhythm. And this regular rhythm has the beauty that it basically elicits no touch response after very few spray pulses. Because this is really a culprit with taste stimulation. When I put a drop on the tongue, it's eliciting a touch response. I can feel it touch my tongue, and our somatosensory system will fire and it will give a response. But I want to study the taste response. So how can I disentangle that? So I silence the somatosensory system on the tongue by applying this regular pulse. So literally the tongue feels numb. And after a few pulses, I can start introducing a taste stimulus. So I have a tasteless liquid, which is indicated here in black, and then a taste stimulus is embedded in this sequence in blue. This really serves several purposes. I have the ability to control my stimulus exactly in time because this pump is super precise and I know exactly when the stimulus is expelled. And also I can measure when it hits the tongue. And it's also spatially precise in a way that if I use a divider on the tongue, like really like just a physical barrier, we can have even lateralized stimulation. We can keep the constant temperature. And because we just spray it into the tongue, so people do not need to swallow, because in order to be sprayed down the tongue, they protrude the tongue a little bit. They don't need to swallow, which does not give us those horrible artifacts that you see here in black in this EEG trace. And it's really unimodal because we silence the somatosensory system. And this technique has really made a big difference for us and allowed us to actually investigate what I'm showing you now. So the signal that you may get from such a stimulation is shown here in an exemplary participant. So on the x-axis, you always see the time axis, and time point 0 is always the presentation of the stimulus. And on the y-axis here, you see amplitude with positive going upwards. And each colored trace is the average of a few repetitions of a given taste solution. For example, the blue trace is a salty taste at an exemplary electrode over the vertex. So, and you can see here already that a lot of stuff is going on. There's a lot of latency differences, amplitude differences, differences between the tastes, right? And this is only one electrode and one participant. So we record from many electrodes and many participants over many trials. So we really have multidimensional data. And the fact that there's literally nothing in the literature actually telling us where to look in the data, standard hypothesis-driven frequentist analysis is really not doing it. So we resorted, so here you can also see it summarized over 18 participants. It looks a little bit tidier here, but still it's very hard to actually decide which condition you want to compare at which time point, etc. So we resorted to a multivariate pattern analysis. And for this, we take the signal from 64 channels that we recorded, all the time points that we recorded in 1.5 seconds. We sampled with 500 hertz, so it's quite a few time points. And we basically use about 90% of the data to train a classifier to discriminate between our different tastes that we presented. And after this learning period, we take 10% of the remaining, so the remaining data for actually testing and see whether there is some kind of pattern, some kind of logic that may not be visible to the bare eye in order to tell what taste has been presented to our participants. So the first question that I had mentioned at the beginning, do those neuronal response patterns contain any specific information for the different tastes? I call them large-scale neuronal responses because we don't know exactly where they're coming from because we are picking up the entire brain here. So in order to do this, we presented a salty, sour, bitter, and sweet taste solution to our participants in random order, many repetitions, many hours. And participants would receive a taste for 900 milliseconds, and then they would be given a pause before they had to say which taste they actually tasted. And we asked the classifier the exact same question. So which taste of the 4 possibilities is it actually? And what you see here is decoding accuracy on the y-axis and again time on the x-axis. And you see here in dark gray the average decoding accuracy in a sample of 16 participants. And what is actually apparent is that we have above chance decoding starting within 175 milliseconds after taste presentation. That means within the EEG signal, we do have information about the taste quality within this time period. And you see a decoding accuracy stays up way above chance. So this information remains available over seconds. So this was actually a very surprising finding First of all, because it's super early, it's super fast. Before, people thought taste would be a very slow sensory system and it would take hundreds of seconds for us to even recognize a taste and respond to it. And here we see now it's actually pretty fast, and it's actually corresponding to the earliest time point where we also see a rise in global field power or electric field current to the taste stimulus. So as soon as we taste basically something, it seems like the brain has the information about what it is that we are tasting. So importantly, because the tastes differ in their hedonic value and their pleasantness, but also in their intensity, it's impossible to match all of those factors. We looked whether intensity shown to the left here and pleasantness, no, it's the other way, intensity on the right, pleasantness on the left, actually correlated with decoding performance, and it did not. So we think it's really not a misalignment, but it's really representing taste quality. But we noticed something else in the data. So here I show you behavioral confusion metrics. And at the bottom, you read the taste that was presented. And at the y-axis, you see the taste that was reported by participants. And what is actually apparent is that when a sour stimulus was presented, in some instances, participants said it was salty. And when a salty stimulus was presented, participants sometimes said it is sour. So they seem to be not quite clear about the difference between the two. And we wondered, well, if they make this confusion, then this should be also mirrored at the brain level. And this is what we looked at. So we now actually trained a binary classifier asking, is it taste 1 or taste 2? And when we compare sour with salty responses, we see no significant decoding performance here. So the brain signal also isn't certain about what it has been tasted. Just to give you another example, if we contrast a salty with a sweet taste, performance is doing pretty well. So it's not the binary classifier by itself. So here we have a very nice correspondence and link between the taste-related decision-making that participants really do and the value, the predictive value that we can take from those brain response patterns. So basically, if we had looked at the brain before, we may have been able to predict those wrong or faulty behaviors. To confirm this further, we correlated those behavioral confusion matrices with the neuronal confusion matrices over time. And it looks pretty much like the first graph you saw, where we have, again, a significant correlation starting within 175 milliseconds between those two, which is, again, confirming what I just said and also indicating that what we measure in the brain is really already reflecting the perception, the interpretation of the chemical stimulus, but not the chemical stimulus by itself. So it's a neuronal perceptual mapping, not chemical perceptual, not a chemical neuronal mapping. So let me sum this up for you. So large-scale neuronal response patterns code taste quality. I hope I could convince you of that. And this happens at the earliest level of the central taste processing cascade that we have been able to measure at the scalp level. So it's basically super fast. It reflects a neuronal perceptual rather than a neuronal chemical mapping. So it reflects what you think you taste, not what you actually tasted. So with that, we went on to actually ask whether those neuronal patterns predict also the speed at which a decision can be made about a taste. And we got inspired to ask this question because reaction times and independently neuronal latencies for taste have been shown to vary between the different tastes. So we thought there could be really a link. It could be the informative aspect in the pattern that the classifier used. So we used a very similar paradigm like the one I showed you before, but this time the task was to press a button as soon as they tasted anything. So as soon as the taste was presented, they were allowed to push a button, and we would remeasure the latencies it took them to do so. So here you just see the raw data, and it looks pretty similar like in the previous study. A lot of latency and amplitude differences between the different tastes. And when we actually look at decoding accuracy for the 4 tastes that is shown here in blue, we see that we have an onset that is a little bit earlier than what we had seen before. Namely, it starts at 130 milliseconds. And I have plotted here the red curve, which is basically the data from the previous study, which is a different group of participants also, where they did not have to make a speeded decision. They could take their time to decide what they tasted. And you see the latency is a little bit shifted. So it's like 40 milliseconds. It does not seem much, but it's a lot for the brain. So there seems to be some task dependency also on the speed of brain processes and when this taste information actually becomes available. So, but let's look at the link between the response times and the brain information about the taste. So here what you see on the left side is again time plotted against decoding accuracy. And here now we have added the time point at which participants pushed the button. So this is indicated by the vertical dashed black line. And in contrast, you see also the blue dashed line, and that is simply cutoffs that we set based on our single trials when decoding accuracy actually reached significance in each subject. And you see there's a gap in between them. That is not surprising. One would think information is first available in the brain before a person is able to act upon it. Also, once information is available, you need a bit of time to actually elicit that response to push the button. So this was expected. However, when correlating the reaction times with the onset of decoding accuracy, we found a positive correlation. I think the plot is not the best. We were actually forced by the reviewer to have an aligned time scale. So it's all like cluttered in one spot, but it's highly significant. And this is for the salty taste, but we can do this for all 4 tastes. And the patterns actually look pretty similar. We find a nice alignment of the link between the onset of the decoding accuracy and the response time of our participants. So actually the timing, and I should say those data were taken at a single trial level. So we would also always compare within a trial the onset of decoding and the given response belonging to this very trial. So the timing of neuronal taste representation predicts the behavioral response times. And again, we looked whether the experienced taste intensity or the taste pleasantness played any role, and we could not find any correlation here between this decoding onset times and those pleasantness or intensity ratings. So what does this add to what we just learned before? So those large-scale response patterns, they predict also taste-related behavior, and that in a goal-dependent manner. So what I mean with that is that it is task-specific. So if you're forced to give a fast response, your brain will also have the information faster available for you to help you do this decision. So it definitely drives your decisions. So let's then look at what I said at the very beginning, where I said, like, it seems like as soon as you taste something, you know what it is, right? That is what the first analysis suggested. But is that really the case? In order to answer this, we derived a somewhat more complex experiment. So we had two different experiments intertwined. In the first part of in one part of the experiment, participants had to make a detection decision. So they were either given water or a taste, and their decision was to basically push a button, which it was. And the second task was a discrimination task. So they were given either of two tastes on each trial, and they had to say which taste it was. For example, sour or salty, or sweet and bitter. Those two could occur in combination. And they were allowed to push the button as soon as they wished, or as soon as they could, better. So they were instructed to go full speed ahead. So here's the behavioral data. So when we look at the salty sour pair, which we looked at together, and you remember people tend to confuse the two, what we see is in the detection task here shown by the blue bars, participants were much faster than in the discrimination task shown in the gray bars. That is not a surprise given the cognitive load. So in the detection task, they had to actually just say, is it water or a taste, whatever taste it was. It's not a difficult ask. While in the discrimination task, they really had to think about which taste it could be. So let's look at the brain level. So here we have again decoding onset latency, the earliest part where we have information about which of the tastes it is. And again, in the detection, we see it's much faster, starts like at 120 milliseconds than it is in the discrimination task. So, and this is not an error in plotting. It's not the same data. It's really behavior on one side and brain activity on the other. So, but we had also a different condition, you know, the sweet-bitter comparison, and there we see a completely different picture. Detection and discrimination latency behaviorally does not make a difference. It does not matter whether you discriminate or whether you detect sweet amongst water or whether you discriminated against bitter. That was surprising. And look at the brain data. The exact same pattern of results, no latency difference whatsoever. So here you can see it all. So what that tells us is that there are some taste-specific sequential, but also parallel processing of gustatory information, which is highly taste-dependent. So let us look at the differences also between those tasks, between discrimination and detection to highlight that a bit further. So here you see the response times versus the onset of decoding times in those correlation plots for the 4 different tastes. And you really see that the detection precedes the discrimination, is shorter and faster, only in the salty and sour conditions, but not in the case of sweet and bitter. That is not a surprise, right? From the graphs, we would have expected that those data points cluster around 0 for sweet and bitter. So here the idea is that detection does precede discrimination. However, if hedonic processing comes at play, comes to the table, that may facilitate discrimination as a parallel process. What do I mean with that? Sweet and bitter, we learned it's very pleasant or very unpleasant. So sour and salty does not have a pleasantness difference in our experiments, but sweet, no matter the concentration, people are gonna love it, and bitter, they're not gonna like in the experiment. So here it's not only about taste perception, but it's really about liking and pleasure. And we believe that those processes, those hedonic processes, kind of run in parallel and speed up the overall response time. That has been recently shown in an animal study. So, well, to my question, as soon as you taste it, you know what it is. Yeah, but only if it's sweet and bitter. So for stimuli that have no particular hedonic value, like sour and salty, this is not the case. So detection and discrimination, they operate sequentially, but also in parallel, depending on the taste quality. And that sums it up to actually being a bit more complicated than we thought, and that we are actually really able to explain. So, but together the data, I think, provide nice amount of evidence that there's a flexible and a goal or task dependent, but also taste specific link between what we see at the brain level and what can be predicted from it about human behavior. And with that, I would like to move on to the last experiment that is not so much about behavior, but about individual differences. And that is really an important aspect in gustatory processing. Because it's a question that has been raised a lot, because I showed you at the beginning that we can learn to appreciate things, or maybe also unlearn to appreciate salt with the dietary change. So one obvious question always is whether the taste processing in lean and obese individuals differ, kind of also with the assumption that their dietary patterns differ in a way. So what did we do to study that? So we had a manipulation of two different tastes. So we had a salty taste and a sweet taste that we presented at two different concentrations. So we did not present those images. Those are just illustrations. And we had a control condition which contained two squares of two different colors and two different sizes. And the size and concentration manipulation basically also modulates the sensory evidence. So a small concentration or a small sized square also means there's little sensory evidence, and if it's larger, there is more available. With that design, we tested 30 lean and 25 obese individuals using our typical taste stimulation protocol. And here is the results. Again, we have decoding accuracy plotted here against time. And the blue curve represents the lean group, and the red curve represents the average of the obese individuals. And the classifier was basically decoding taste and answering the question, is it sweet or is it salty? And what we see is that early on, taste information emerges evenly rapidly in both groups, and it does not differ at all. That was a surprise because some people said that obese may actually experience the taste early on already differently. However, what is striking is this very late difference. So after 1 second of processing, this is very late in perception terms. So here we have a difference between the groups that indicates that obese participants lose this information about the difference between the tastes earlier than lean individuals do. Because remember, the curves reflect the coding accuracy between the two different tastes. So it looks almost like the taste information is fading from the obese brain. Importantly, when we do the very same analysis for our visual stimuli, classifying green versus blue, we do not find any group difference whatsoever. This is a very important thing to do because whenever you compare different groups, there may be a lot of also physical differences, like the connectivity at the skull level, if you have a huge weight difference, for example, but that does not seem to be the case here. And as a next step, we were wondering whether we can even go as far to classify the membership of the group. So basically here we're not classifying tastes anymore, but we're classifying people, asking here for the different tastes plotted separately, whether we are having looking at a brain response from a lean or an obese individual. And what you can see here is particularly in this later time period that we saw, where we saw the group differences. Here we also see significant decoding differences for the group membership. And again, when we look at the very same data, I just plotted it next to another, we do not see those group decoding at all when we look at the green or the blue squares that we are presented. So it is really a differentiation of the neural representation of the taste, but not the color stimuli, that allows us to differentiate between the individuals. And whenever you look at obese and lean individuals, one always have to take that with a grain of salt and be careful, because there are a lot of very healthy overweight participants, and we don't have that information. So it is a very, very coarse categorization that we're doing here, and that requires quite some more investigation also into their specific dietary habits. So what I would like to conclude from this is that those large-scale patterns can differ between individuals. In our case, it was obese and lean. And so that indicates that there is some individual variability that may have something to do with the physical status, something to do with the behavior over the past years. But from our study, this is very hard to tell, and I don't want to really go there and make that statement. So it seems like we lost one result here in the slide. So I would not change the overall conclusion from that, and hope I could show you with the series of studies that we do have evidence for a flexible, a task-dependent and taste-specific link between taste-related behavior and the brain response patterns. And here they come. And with that, I would like to thank the people who did most of the work, my students, Raphael Wallroth and Richard Höschenberger, and our technician, Andrea Kaczak, who did a lot of the work, and my collaborator, Nico Busch, and of course, also those who gave us some money to run those studies. And with that, I thank you for your attention, for your patience, and I'm very much looking forward to your questions. Thank you.
Thank you very much, Kathrin. I think the question session is opened. Are there questions to this talk? At the moment, we don't have any in the chat.
I see one hand.
Okay. Oops. Okay.
I just want to ask, because I just noticed that the classifiers are not doing like a really, really good job. And I was wondering if this is because you are looking at the whole brain. And my question is like, why don't we know that there's some specific area for the taste where you can focus and decode the neural data from there? It's just maybe a naive question, but.
No. It's a very important question. So what we are recording is a super noisy system. So we are recording, first of all, the entire brain, while probably only a fraction of neurons are processing what we are interested in. And I mean, the participants are doing things, right? They're thinking about dinner plans. Maybe they just had a fight with somebody. Those thoughts are really making this very noisy. And it's a very obvious suggestion to just focus on one part of the brain where we think, most of the processing is taking place. However, this is hard to do with EEG. It can be done with fMRI. However, because you have a great spatial resolution, but no temporal information. If I were to use those EEG data, I could make a source reconstruction and try to pinpoint where the data is coming from. But then with all the variables, right? I mean, I'm going to have so many time points, and then I'm going to add to my sensors, which are now 64, I'm going to add dimensions maybe to a couple of thousand points that define my source space. I would need so much data to do a meaningful decoding. And those experiments take already hours. You would have to record for several days probably to do that, but I would love to.
Okay, thank you. Wilhelm, will you ask your question?
Yeah, I think it's related. So the question is, so whether you, let's say, looked at the support of the classifiers more thoroughly. So what channels are, let's say, more active, more, let's say, where is the classifier, the distinction, let's say, concentrated, and whether that changes in time, for example. And this could be a hint, for example. And then a related question to that would be, do you see a distinction between these, let's say, kind of signals and, let's say, other cognitive tasks? Like if you look at EEG data and let's say people are listening to music or something, visual stimuli or something like this. So is there some characteristic differences in the EEG signals that you have observed?
So your first question, if I got it right, so what we did was we would reduce the number of electrodes. We would look at 1 electrode, 2, 3, 4, and we would just randomize this to have every possible combination of electrodes to maybe get rid of those who don't add information to our analysis. That did not work at all. So as soon as you remove some, you lose even more of your decoding accuracy. And your second question. So when I originally came from vision science. So when I recorded my first taste responses, I thought I programmed the experiment wrong, because to me, it looked like a beautiful visual evoked response, like the first figure that I showed you. And so we have very prototypical peaks and troughs that are closely related to a visual response, I would say. Like an auditory response has much more smaller or somatosensory and faster responses. That said, when we look at the topographical distribution, like where on the scalp the signal is coming from, that's entirely different. But I think it's fair to say that the sensory systems share also properties in their evoked response, in that they have a sequence of peaks and troughs of different components. And I think it's also fair to say that those very early, the first components, whenever they occur, right, that varies from sense to sense. They reflect more sensory aspects that are closely linked to the physical or chemical nature of the stimulus, like the concentration, the size of an image, the loudness of a sound, for example, while the later aspects of this signal reflect more cognitive aspects related to memory, related to evaluation. Do I like this? Can I name it? Things like that. And those components, they should be really universal across sensory systems because they are cognitive.
Okay, are there more questions? I have a tiny one. At the beginning, when you described your experiment, you said that you would like to partition the tongue. Why would you like to do that?
So partition sounds almost painful. A crazy thing actually in gustation is that we don't really know where the fibers project to. So we know, for example, in somatosensory systems, the input from the left side of the body is projected eventually to the right side of the brain and vice versa, right? We know this about hearing, we know how it works in vision, but we don't know how it works for taste. So we don't know whether the fibers cross, at which level they cross, and to which degree they cross, because in visual, we also have just a partial crossing. So in order to investigate that, we needed to stimulate the tongue of the right side and the left side of the tongue separately, because they are innervated by lateralized nerves. So, and with that, we could get a handle on where information may go.
Okay. Okay. There's a question in the chat. Cheng Li said, very nice work. Are there analogous experiments with more invasive recordings of trained primates who can distinguish taste? Say electrophysics recordings focused on gustatory and olfactory cortex?
Not in recent decades. I think the last invasive recordings with primates were done in the 80s. So there are a few groups, I think in Tübingen, who do fMRI experiments on taste with primates, but actually the animal model that is closest to the human is actually the mouse or the rat. So I'm typically relying really on rodent data, because that gives us also the latest data. And there we have recordings from gustatory cortex, from primary gustatory cortex, where we see almost identical data of what we see here at the scalp level, but with a better data quality, because all this noise and also all this cognition that we have with the human is so much lower when you have a direct measure from a certain brain area.
So there you would get the chemical space as well, or at least partially.
Yeah, those questions are actually a little bit trickier to ask in an animal, to actually answer in an animal, right? Because you can look at behaviorally, you can look at approach and avoidance, and you can deduce if they continue to consume the sample, they must like it and they must recognize its sugar. And if they do not continue to consume or even show an aversive behavioral response, they must have recognized it's bitter and it's aversive. That's a bit of a stretch and a bit of a different quality of decision than asking a human which of the four tastes was it, right? So with humans, we can get much more precise and direct reports of what they experience. With animals, we don't get that.
Okay, thank you. So if there are no more questions, I'll thank you again for the very nice talk.
Thank you for having me.